The Niger River does not rush here. It spreads. It flattens. It moves the way a people move when they have nowhere left to go and everywhere still to claim. In what is now the inland delta of Mali, that vast, shimmering floodplain where the river loses itself across 20,000 square kilometers of grass and mud and ancient light, a woman is dying. She is not old. She is perhaps 32, 33 years into a life that has been defined by two things: the smell of wet sorghum drying on flat stone and the sound of the Mande language moving through her household like breath. She has never had a name in any history book. No pharaoh commissioned her portrait. No merchant recorded her trade. She belongs to no dynasty that archaeologists have yet dignified with a proper noun. It is approximately 2000 BCE. The early Bronze Age is reshaping everything north of the Sahara, but here, below the Great Desert, along the river's soft interior, her people are doing something older and more patient. They're building a genetic army. She does not know this. She knows only the heat, the specific lung-pressing heat of the Sahel in the dry season, and the taste of iron in her mouth, and the faces of the people leaning over her. Her petrous bone, the hardest bone in the human skull, the small pyramid-shaped fortress that houses the inner ear, is doing what it has always done. It is holding everything still. It will hold everything still for 4,000 years. For most of the 20th century, ancient DNA research in sub-Saharan Africa was, scientifically speaking, almost impossible. Not difficult, not challenging, impossible, and the reasons were brutally specific. DNA degrades through a process called hydrolysis and oxidation. Heat accelerates both. Humidity compounds the damage. The equatorial and sub-Saharan regions of West Africa, precisely the zones most critical to understanding Black American ancestry, sit between the 10th and 17th parallels north, where average annual temperatures range from 25 to 38 degrees Celsius and humidity during wet seasons regularly exceeds 80%. Under these conditions, the double helix breaks down. The base pairs separate. Cytosine converts to uracil. Within a few centuries, in most burial contexts, the DNA is simply gone. Not degraded, not fragmentary, but chemically absent. This wasn't a funding problem. It wasn't a political problem, though the politics were never innocent. It was thermodynamics. The equatorial sun was functioning as a perfect evidence destruction machine, operating on a 365-day schedule for thousands of years before any scientist arrived with a sequencer. The consequence was staggering and specific. While researchers successfully extracted ancient DNA from 45,000-year-old Siberian bones, from 5,300-year-old alpine mummies, from 3,500-year-old Egyptian teeth, the genetic past of West Africa, the direct ancestral homeland of 30 to 40 million Black Americans, remained a void. Not a mystery. A void. There was nothing to sequence because there was nothing left to find. By 2015, the ancient DNA map of the world had a hole in it shaped exactly like the Middle Passage's other end. The solution came from anatomy. In 2015 and 2016, researchers including ancient DNA specialists at Harvard Medical School's Reich Lab and collaborating teams at Johannes Gutenberg University in Mainz, Germany, made a methodological pivot that changed everything. They stopped drilling into the densest long bones, the femur, the tibia, and started focusing on a single structure, the os petrosum, the petrous bone. This small, wedge-shaped portion of the temporal skull, less than 4 centimeters long, contains the hardest substance in the human body. Its density, approximately 2.4 grams per cubic centimeter, is higher than almost any other skeletal material. More critically, the inner ear canal it encases is naturally sealed, protecting the DNA of cochlear cells from environmental exposure. In a 2015 study published in Nature, researchers Pinhasi, Fernandez, and colleagues demonstrated that the petrous bone yielded ancient DNA at concentrations between 44 and 183 times higher than other skeletal elements from the same individuals. 44 to 183 times higher. That number is not a marginal improvement. That is the difference between silence and speech. The second breakthrough was chemical. A technique called UDG treatment, uracil DNA glycosylase, allowed researchers to identify and partially repair the cytosine to uracil damage that heat causes, essentially letting scientists read degraded sequences that would previously have been dismissed as noise. Combined with next-generation sequencing platforms capable of reading millions of short DNA fragments simultaneously, fragments as brief as 35 base pairs, and cross-referencing them against modern reference panels, ancient genomes from humid tropical environments became, for the first time, retrievable. In 2023, a landmark study published in Nature by a team including researchers Lipson, Sawchik, Olalde, and 39 co-authors examined ancient DNA from 91 individuals across 14 sub-Saharan African archaeological sites. Several of those individuals came from burial contexts in present-day Mali, Burkina Faso, and the Niger inland delta, sites dated between 4,200 and 1,800 years before present. The petrous bone made it possible. The UDG treatment made it readable. And what those sequences contained rewrote the Ancestry conversation entirely. The 2023 study and the cluster of research that followed it through 2024 produced findings that were simultaneously confirming and astonishing. The ancient individuals recovered from Malian burial sites dating to approximately 2000 to 1800 BCE showed a genetic profile that researchers designated as a deep branch of the West African ancestry clade, a lineage distinct from the East African and Central African genetic clusters that dominate older ancient DNA literature. Using principal component analysis and admixture modeling at K equals 8, these individuals clustered most tightly with present-day speakers of Mande branch languages, the language family that includes Mandinka, Bambara, Sonic, Dayala, and over 40 related tongues. The divergence time between this ancient Malian cluster and the closest East African ancient lineages was estimated at approximately 60,000 to 75,000 years before present, consistent with the population split that occurred as anatomically modern humans dispersed across sub-Saharan Africa following the major southern African expansion. But the critical number, the one that connects ancient Mali to living people, is this. When researchers modeled the genome of a present-day Mandinka individual from the Gambia against the 4,000-year-old Malian ancient genome, the genetic continuity was approximately 76 to 84%. 3/4 of the Mandinka genome has not moved, not mixed, not shifted in 4 millennia. The Mandinka people of the present-day Senegambia region carry the genomic signature of the Niger inland delta, intact across 4,000 years of climate change, empire collapse, trans-Saharan trade, and the violence of the Atlantic world. The ancient individuals also showed no detectable Eurasian or North African admixture, no signature of the Bronze Age steppe migrations that transformed European and Middle Eastern populations during the same period. The Sahara, in this era, was functioning as a nearly complete genetic barrier. What was south of it stayed south of it. What was north never came south. The West African interior was developing its genomic identity in isolation and in depth. Further, a 2024 companion study examining ancient DNA from Iron Age sites in present-day Senegal and the Gambia, sites dated to approximately 900 to 400 BCE, showed the same ancestral profile propagating forward in time. The Mande-associated West African clade was not a local burial population. It was a continental anchor. This is where the science stops being abstract. If you are a Black American whose ancestry traces to the Senegambia region, the zone that historical slave trade records identify as the origin point for approximately 16 to 24% of all Africans enslaved and transported to North America, then what was just described is not ancient history. It is your genome. The Transatlantic Slave Trade Database, compiled by historians Eltis, Richardson, and their collaborators, documents that between 1600 and 1867, approximately 388,000 to 450,000 enslaved Africans were brought directly to what became the United States. Of those, the Senegambia region, modern Senegal, Gambia, Guinea-Bissau, and coastal Guinea, accounts for a substantial documented corridor. South Carolina's rice plantations specifically sought enslaved people from this region because of their agricultural expertise. The Gullah Geechee communities of the South Carolina and Georgia Sea Islands are, in part, a direct demographic continuation of forced Senegambia migration. When you take a consumer DNA test today and it returns a result that says Senegal or Mali or Ivory Coast/Ghana, that result is being calculated against modern reference populations. But those modern reference populations are, as the ancient DNA now confirms, themselves direct descendants of lineages that existed in the same geographic zones 4,000 years ago. Your DNA test is not just pointing you to a country, it is pointing you to a temporal depth. When your result says Mandinka-associated ancestry, the genome it is detecting in your blood is genetically continuous with a woman dying on the bank of the Niger inland delta in 2000 BCE. The Middle Passage tried to erase that continuity. It erased names. It erased languages. It erased kinship structures and religious calendars in a specific way a particular grandmother nodded her head wrap. But it could not erase the petrous bone. It could not erase the cochlear cells. It could not reach inside the genomic record and perform the same destruction the heat had spent centuries attempting. Your DNA held. If your 23andMe or Ancestry DNA result includes any West African percentage, and for most Black Americans that number sits between 52 and 83%, you are carrying ancient Malian, ancient Senegambian, ancient Niger Delta DNA that predates the Roman Empire, that predates the pyramids at Meroe, that predates every dynasty the Western world has decided to teach in a classroom. You were never disconnected from African antiquity. You were always genetically inside of it. The only thing that was missing was the science capable of proving what the bones already knew. Here is the finding that went beyond the headline. When researchers examined the ancient Malian genomes for evidence of population movement within West Africa, using a technique called F3 statistics, which measures shared genetic drift between three populations simultaneously, they found something that complicated the narrative of West African genetic isolation. Approximately 1,200 to 1,400 years ago, between 600 and 800 CE, there is a detectable pulse of Central African ancestry, specifically a genomic signature associated with Niger-Congo Bantu expansion lineages, appearing in the ancient DNA record of Western Sahelian populations. This is not Eurasian admixture. This is not North African admixture. This is African movement, west to central to west, a genetic dialogue happening entirely within the continent. The timing corresponds almost exactly to the height of the Ghana Empire, the Ouagadougou state, the first of the great West African empires, centered in the region between modern Mali and Mauritania, which dominated trans-Saharan gold and salt trade from approximately 600 to 1200 CE. At its peak, the Ghana Empire was integrating population from the Senegambia in the west to the Niger Bend in the east and maintaining long-distance commercial networks that stretched south into the forest zones where Bantu language communities had been spreading for centuries. The ancient DNA is recording the Ghana Empire's population dynamics. It is recording the movement of enslaved workers, traders, soldiers, wives, and merchants across a vast imperial geography, not as an abstraction, but as a genomic event, measurable in base pairs, datable to within a few centuries. For Black Americans, this layer matters because it means your West African ancestry is not a simple, single-source signal. It is a layered signal, Monde core ancestry sitting atop an older Sahelian substrate, with a 1,200-year-old pulse of Central African lineage woven through it, all of it compressed into the same chromosomes. The Mandinka person your DNA points to was already a synthesis. West African genetic identity was built through internal African movement, internal African empire, internal African complexity, long before any European ship appeared on the horizon. That complexity belongs to you. It always did. Return to the Niger inland delta. Return to 2000 BC. Return to the woman dying in the dry season heat with the taste of iron in her mouth and the sound of Monde moving through her household. She was never recovered by archaeologists with brushes and grid paper. Her specific burial site has not been assigned a catalog number in any published database. She is composite. She is every unnamed ancient woman whose petrous bones survived in the Sahel's deep soil, in the sealed chambers of inland delta burial contexts, in the particular luck of geological preservation that kept a few cochlear cells intact across 40 centuries of sun. But her bones prove something that no archive, no plantation record, no census roll, no slaver's manifest could prove or disprove. That the genetic line running from the Niger inland delta to the Senegambia coast, to the holds of ships, to the rice fields of South Carolina, to the blood of living Black Americans in Atlanta and Houston and Baltimore and Oakland, that line did not break. It bent. It was compressed into chains, and then into containers, and then into the bureaucratic language of property law. It was renamed and redocumented and stripped of its geographic specificity by the machinery of the Atlantic world. But the petrous bone is not a document. It does not respond to renaming. It does not degrade under the pressure of forced forgetting. It held the sequence in the dark for 4,000 years, waiting for a drill bit thin enough and a sequencer fast enough and a scientist patient enough to finally ask the right question of the right bone. And when they asked, the answer was unambiguous. The Mandinka bloodline did not survive the Middle Passage despite everything. It survived because of what it was built from. 4,000 years of continuity, accumulated before the first ship was ever constructed, deposited in the hardest bone in the human body, carried forward in the blood of people who were never, not for one generation, not for one century, actually erased. The bones remembered everything.